Photosynthesis and energy transfer. Photosynthesis can be summarized very easily as water plus carbon dioxide in the presence of light produces sugar and oxygen. Photosynthesis is actually a series of reactions which are driven by light energy, and it can be divided into two stages. The first one being the light dependent stage. This depends on the presence of light. And number two, the light independent stage. Of course, this does not depend on light, however, it does rely on substances produced in stage one. Both stages of photosynthesis take place in the chloroplasts in plant cells, but they take place in different parts of the chloroplast. The chloroplast is a double membraned organelle within the plant cells. Inside the chloroplasts, there are a series of interconnected disc-shaped structures. These are called grana, and they're involved in the first stage of photosynthesis. The chloroplast is also fluid-filled. The fluid contains enzymes involved in the second stage of photosynthesis, and this area is called the stroma. So we begin with stage one of photosynthesis, the light-dependent stage. When light is absorbed by a molecule of chlorophyll A, some of the electrons become excited in the pigment molecule. The electron now has a higher potential energy, and it would normally revert to its lower energy state, releasing the energy as heat and light. However, it's prevented from doing so, and it's captured by the primary electron acceptor. The electrons then travel through an electron transfer chain which releases energy. This is used to generate ATP by the enzyme ATP synthase. It's also used to split water into oxygen, which is released, and hydrogen, which becomes bound to the coenzyme NADP, which acts as a hydrogen acceptor to form NADPH. And this is stage one, or the light-dependent stage of photosynthesis. And this is summarized on the next slide. Here we have a close-up of one of the membrane structures within the chloroplast, or the grana. Here is the light harvesting complex and the primary electron acceptor. There's a molecule of chlorophyll A. Here's the electron transport chain, and here's ATP synthase. Light excites the electrons. The electrons are not allowed to fall to a lower energy state in the chlorophyll. Instead, they are passed along the electron transport chain. This causes water to be split, releasing two electrons which replace those lost by chlorophyll. Oxygen is given off. As the electrons pass through the electron transfer chain, hydrogen ions are pumped into the inner space. The high concentration of hydrogen ions now begin to flow out, driving ATP synthase and producing ATP. The energy from the electrons is now used to join NADP, a hydrogen acceptor, with hydrogen to form NADPH. And here is a summary of stage one. As light energy is used to split water, the first stage in photosynthesis is also known as photolysis. So we come to stage two, the light-independent stage. The light-independent stage also takes place in the chloroplast. It does not require light, but it needs what is produced by the light-dependent stage, which is the NADPH and ATP. Like the first stage, it is also a series of enzyme-controlled reactions, and it forms a cycle called the Calvin cycle. It's also where carbon dioxide is used and where sugar is produced. In the carbon fixation stage, or the Calvin cycle, the enzyme rubisco fixes carbon dioxide by attaching it to ribulose bisphosphate, or RUBP. The 3-phosphoglycerate, or 3PG, produced is phosphorylated by ATP and combined with hydrogen ions from NADPH to form glyceraldehyde 3-phosphate, or G3P. G3P is used to regenerate RUBP and for the synthesis of glucose. And that can be summarized like so. The glucose, or sugar produced by photosynthesis, can be used in cellular respiration, or can be involved in the growth and reproduction of plants and production of DNA, protein and fat molecules. It can also be converted into complex carbohydrates and stored as starch, as an energy reserve, or used for the production of structural cellulose in cellulose cell walls. In terms of plant productivity, biomass of a population is the total mass of organic material, and it's usually measured in dry mass, as the water content of living organisms can vary. Assimilation in plants is the process by which food produced by photosynthesis is incorporated into complex constituents of cell parts such as protoplasm and cell walls, and this results in the increase of biomass of the growing plant. The rate of photosynthesis is affected by a number of environmental limiting factors, and these include temperature, light intensity and carbon dioxide concentration. This means that when either light, temperature or carbon dioxide are in short supply, they affect the rate of the reaction, in this case photosynthesis. But it should be noted that at very high temperatures, no photosynthesis will occur due to the fact that enzymes are denatured at high temperatures. In this graph showing the action of limiting factors on the rate of photosynthesis, we can see that in the first part of the graph, the limiting factor is CO2 concentration, but after the line split, in the second part of the graph, the limiting factor in the central stage is light intensity, but the limiting factor for the final part of the graph, with the information that we have been given, is currently unknown, but we can assume it may well be temperature.